presenting the result of the unknown bacteria by group 4. After 24 hours of incubation, it's time to check the results of our bacterial test. First is the catalase. The catalase test is positive, indicating the bacteria produce the enzyme catalase. Next is the coagulase. The bacteria are coagulase negative, suggesting they do not produce the enzyme coagulase. This differentiates them from Staphylococcus aureus, which is coagulase positive. Now we have the MSA, or the mannitol salt agar. The colonies are pink, indicating that the bacteria do not ferment mannitol. This points towards Staphylococcus epidermidis, as it is known for its inability to ferment the mannitol, unlike the Staphylococcus aureus. Next is the BAP, or the blood agar plate. The colonies exhibit beta hemolysis, meaning they completely lyse the red blood cells, creating a clear zones around the colonies. Next, the CAP, or the chocolate agar plate. The colony shows small to medium-sized, gray to white colonies, which indicates that it might be a S. epidermidis. Now the MAC, or the McConkey agar plate. There is no growth on MAC, and this indicates that the bacteria do not grow in the presence of bile salts and crystal violet, suggesting they might not be a gram-negative enteric bacteria, which typically grow well on this medium. Then next is the EMB, or the eosin methylene blue agar. There is no growth on EMB, and this suggests that the bacteria do not grow in the presence of eosin Y and methylene blue dyes, which are used to select the gram-negative bacteria and differentiate them between lactose fermenters and non-fermenters. Now let's proceed to AST, or the antimicrobial susceptibility testing. And first we have the nitrofurantoin, and the bacteria are susceptible to it, and it indicates that these antibiotics could be effective treatment option. Next is the clindamycin. The bacteria is susceptible to clindamycin, indicating that this antibiotic could be effective treatment option. And lastly, the other antibiotic discs that we use for antimicrobial susceptibility testing are bacitracin, optochin, penicillin, and ampicillin, that shows a no zone of inhibition, which means that the bacteria are resistant to these antibiotics. To conclude, these tests together provide a comprehensive profile of the bacterial species present, helping to accurately identify and understand their metabolic capabilities. Therefore, we conclude that the bacteria is the Staphylococcus epidermidis.